Thank you for having me. Yes, my experience in the age of AI, and specifically my experience of drug discovery in the age of AI. Oh, there it is. So I'll start with a disclaimer. Yeah, so it's a quote. Yeah, so basically, AI is not magic. And although it can do magical things, yeah, so I have a new laptop, and I asked it, Copilot, to draw a molecule out of a hi-hat, and this is what happened. Well, this is the second attempt. The first attempt, the molecule was garbage, and I said, okay. Now I provided it the structure of a Viagra, sildenafil, and I said, use this structure, and then it came up with this. It looks a little bit more like a molecule, but yeah, it's not. Yeah, it's not correct. And if you look at the Q&A slides, there are a few dodgy structures on there as well. So homework for you. There we go. So drug discovery. I'm assuming not everyone works in pharma. So just 2 slides. Where do drugs come from? And I'm talking about the legal variety of drugs. Yeah, and sometimes you hear that 1 in 10,000. Yeah, so people imagine this big funnel, and you throw 10,000 compounds, chemical structures, chemical compounds in through a whole range of filters and assays, and in the end, and screens, and in the end, one drug pops out after about 10 years and 1 billion, and that's your drug. Well, that's not really what's happening. Yeah, so the top bit is not correct. That's the drug discovery phase, where you find your candidate, and I will come back to that later. It's the second part, yeah, the clinical phase. There, your candidate is fixed. You have one molecule, and you put it through a lot of tests, yeah, animal tests, human tests, to see if it is truly a safe and commercially viable drug. But what happens in that first bit? Yeah, so what really happens until recently, you start your process. This is an iterative process. We go through it multiple times. You start with something called a hit. Yeah, it's a molecule that can come from various sources. It's something your competitor has published, or you've done a high throughput screen, etc. And you think, okay, how can I make this better? Yeah, so you come up with lots of ideas. Yeah, and that can go from tens to hundreds to thousands. Yeah, in principle, there is no upper limit. There is a limit. Yeah, you have to make them. Yeah, these are structures that don't exist. Yeah, you have to synthesize them. Yeah, proper chemistry, and then you have to test them. Yeah, in petri dishes or other assays. Are they any good? Yeah, so there's the chemistry and biology. And so you go from many ideas to tens, maybe hundreds that you actually make. Look at the results and you say, okay, am I done? Do I have my candidate? Well, in all likelihood, you don't. Yeah, but you learn from what you've done. You analyze, you learn, have new ideas. Yeah, and so the cycle continues. And typically, yeah, you do this 20 times, 50 times, depending on how much money you have and patience, until you're done, or you think, well, I give up. Yeah, those are the 2 options. But once you have your drug candidate, it goes into the funnel, and that's how hopefully in the end the drug pops out. Yeah, so traditionally when I joined the pharma industry in 1999, yeah, those ideas came from medicinal chemists, yeah, because they are the people who actually know how to synthesize molecules. Yeah, and they first of all look at, this is what we have, how can I modify it? Yeah, that's chemical knowledge. But now AI, you know, what does AI do in this picture? Yeah, so first of all, the generative idea, the generative AI. Yeah, so instead of a human coming up with ideas, you can use multiple methods to come up with ideas. Yeah, instead of human looking at the ideas like yes, no, you can apply a model. And the other thing is, it's called active learning. Yeah, how can I be smart in selecting which compounds to make? Yeah, if I make 10 compounds and they're all very similar to each other, You can argue you're basically asking the same question 10 times. Yeah? So your answer is more, has a higher certainty, but you're only answering one question. Yeah? So if you make them slightly more diverse, you can maybe answer more than one question. Retrosynthesis, yeah, AI has had a tremendous impact in basically making the recipe. So this is the structure I want to make. How do I make it? And again, that is chemical knowledge embodied in a model that tells you, well, you go to vendor A, buy this reagent, go to vendor B, buy that reagent, run this reaction, and the product you run another reaction, and there is your product. Yeah, so that synthesis planning has become that so much better with AI. Yeah, that really changed things. And the other thing is robotics. Yeah, we've seen already examples, and I think we're going to see more of them. Yeah, instead of humans doing synthesis, have robots do them. Yeah, so and two more, decision algorithm. Yeah, so are we done? How are we doing? That's again something where AI can help, although that's a bit in its infancy. And then learning is now updating your models, your machine learning models. Yeah, so here a bit more text. Yeah, so multiple ways to generate new structures. You can do it from scratch. Yeah, large language models generate a new structure from scratch, or you have like genetic algorithms that take an existing and transform it. And yeah, Biovia has a product for that, I have to say, that works well. I don't think it's the biggest bottleneck. Yeah, the bottleneck is more like I have too many ideas than too few. Active learning, choosing which subject to make is crucial. Yeah, that's still ongoing research. That was largely my research. Machine learning, and again, thanks to Biovia and a predecessor company, Cytologic, yeah, they really democratized making machine learning models. Yeah, it's not a specialized skill anymore. Those small, we're talking really small models, because you only make tens to hundreds of compounds. Yeah, you're not training on the internet, you're training on the data you generated yourself. That's tiny. And most of them work. Yeah, if you pick a sensible one, most of them work. And none of them will be great because you train on tiny, tiny data. Yeah, so it makes sense not to spend too much time on here. Yeah, I said AI retrosynthesis really makes a difference. Robotic synthesis hopefully will, but the logistics of getting all the reagents and catalysts and all the other stuff there is still slow, yeah, because the supplier might be in China. And finally, yeah, it's easy to recognize when you have a candidate molecule. Yeah, that's the one that stands out. But it's less easy to recognize when do I stop my project? Yeah, because there's always that hope that the next round will do it. It's like the gambler's addiction. Pragmatically, most projects just set a deadline. You have 1 year, or you have so much money. That's it. Yeah, but that's rather artificial. Yeah, so the whole business case. Yeah, why do we do this? Yeah, you get if you do this well, you have fewer of those cycles to get to a candidate. You need to make fewer compounds, and they are expensive to make, and hopefully also faster. Yeah, if you design them well, that are easy to make. So faster, cheaper, better. Yeah, that's the whole bag of gold at the end of the rainbow. And there's still humans in there. Yeah, nothing of this is perfect. So you need a guardrail. But also humans are, I would say, better at strategy. Yeah, with drug discovery, you always have multiple problems you want to solve. Which one is the most important? Yeah, at this moment, it's hard to beat human strategic thinking on that one. And sometimes there's external knowledge that's not in your model. Like your competitor has done a phase 2 trial and it failed. Yeah, so that might invalidate your approach. Maybe you should stop. Yeah, that's not in your model at all. So that is what humans definitely contribute and will contribute. So there was a fair degree of hype. Yeah, damned if you do, damned if you don't. That was the dilemma we faced when we founded our company. We're looking now on the right-hand side. Yeah, we were chemists, we were biologists, yeah, we were domain specialists, but we were not machine learning specialists, we were not AI specialists. Yeah, we used existing software off the shelf, and to some extent, some of them we used as a black box. Yeah, we knew they worked, but not really the mathematical nitty-gritty. Yeah, so let's stick to what we really know. Yeah, okay, you get feedback. If you don't use AI, you're not serious. Yeah, because on the other side, all our competitors were calling themselves AI companies, and we thought, maybe we are. Yeah, maybe we are an AI company. Let's do it. And then people tell you, yeah, you're only contributing to the hype. But I think in this context, it's important to know who said what. Yeah, so the naysayers, they were competitors, or former colleagues who could have started the company but didn't. Yeah, so the bystanders. And the other remark was from investors and potential customers. Yeah, so who wins? Yeah, that's obvious. Yeah, if you want to run a company. Yeah, but then the crash. Yeah, oh no, sorry, this is the hype. 2017, we were 12 people. And there was this website. It's Money Morning. I think it's a stock picking website. And the guy featured us. And yeah, basically, it was like hype on steroids. Yeah, it was almost funny to read how at least one person on this planet interpreted our work. And basically, he said, well, yeah, Accenture, we were a private company at the time. You cannot buy the stock, but they work with Sanofi and GSK, so buy those company stocks, and you will see an uptick because they work with us. And I don't know if anyone of Sanofi is here in the audience, if you did see an uptick in your stock price, but yeah, it's big companies don't move that quick, yeah, to say the least. That is nonsense. Yeah, so and now we're back to earth, hard. Yeah, all the literature out there, also in popular literature, newspapers, daily newspapers, like this doesn't work. There are no drugs, and yeah, the stock market hasn't been friendly either. Yeah, this is us and competitors of us, all minus 80% in a couple of years. Yeah. So we've been here before, boom, bust, boom, bust, boom, yeah. But maybe there is a boom, yeah. Some people think so, and maybe we're going up again. Yeah. So I see this like gravity. Yeah. I may not like it, it's there. Yeah. You have to deal with it. So sometimes people like what you do, sometimes they don't, and there's nothing you really can do externally. But still, yeah, we wanted a drug discovery companies. Where are those drugs? Yeah, and basically, if people had thought about this, yeah, this should have been known, and they could have known this, and they should have known this. Yeah, so if you make the drug discovery phase faster, it doesn't necessarily mean better. Yeah, if your hypothesis is still the same, you get to the answer quicker. But if your hypothesis is wrong, it's still wrong. Yep. Other thing is even the startups with the most money, you don't have enough for a phase 3. Don't have enough for maybe not even for a phase 2. So you have to collaborate. Yeah, and then it becomes tricky. We walking through treacle again. Yeah, so that it becomes much slower. And raise the money. The stock is down. Raising money is impossible. Yeah, so we're a bit stuck, we as in the discovery-based AI discovery companies. Yeah, so and if you look at the current front runners, yeah, the ones that are in phase 1 and 2, they are the first attempt, yeah, not the best attempt. They were our first efforts. Yeah, and despite all of this, there were some successes. Yeah, so and this is probably as good as you could have expected it to be. Yeah, so on the right-hand side, you see some timelines and how much money is involved. This takes time. Yeah, we could not have been there yet. Yeah, so what did I learn running an AI company, a startup? Growing pains. Yeah. Well, and I will go through these points. Try to do this quickly because I'm already running out. Well, basically, expectations. Yeah, we are a bit guilty. Yeah, we had initial successes on a shoestring. Then you talk to collaborators, potential collaborators, investors, who only want to hear our success stories, yeah, not the ones that failed. We need those collaborators and investors, so we only tell them the success stories and say, yeah, we've done it once, now we can do it 100 times. Yeah, so what happens in all collaborations, we got the most difficult target, and we got huge amounts of money, and I think too soon, too much, and it was in the middle of a pandemic. Yeah, so we quadrupled the company during a pandemic, but that is hard. That was really hard. And you get these kind of cultural differences where suddenly we had proper AI people, and they not only used different words for the same things, but had a completely different way of thinking. Yeah, so black and white versus fuzzy sciences like chemistry and biology, where not all data are that reliable. Yeah, so how to deal with uncertainty, and that works the other way around. Chemists had to work with models, and again, yeah, the models don't always work. Yeah, so how to keep working with them if it failed 3 times. Yeah, so how to keep the credibility up. And then we had people who had already a pharma career behind them. Yeah, they were used to certain ways of working, and instead of becoming part of a startup, we ran the risk of becoming a small, big pharma company, yeah, doing the same things. And yeah, hire people from academia these days. They want to publish and open source everything. And if you're not careful, that includes the company IP. Yeah, so you have to be a bit careful with that as well. But yeah, the real bottleneck in all of this is here. Yeah, making these compounds and screening them. The wet lab part is really expensive and slow, and that doesn't really change that much. Yeah, and it's like the Olympics. Yeah, you have multiple competitors, and there is no second place. Yeah, that's like the Nobel in that aspect. So I will skip the rest of the slide. But basically, you have to learn fast. Yeah, so how to make those experiments count. And we had already a speaker with a database of failures, so that's what you need to do. You need to fail a lot. Yeah, and this is an example from baseball, where the best baseball player of all time was also the worst in strikeouts. Yeah, so he was both the best and the worst at the same time. And that is because they're correlated. Yeah, if you don't try to strike a home run, or if you do try to strike a home run, you will strike out. Yeah, so you will be sent away without success. And there is a correlation, and the same is with drug discovery. Yeah, so if you play it too safe, bottom left, yeah, you make compounds that are very close to your current best, they will be similar. Yeah, and if your current best is not good enough, the next one will not be good enough either. You really have to try something else. Yeah, and if you do random things, you enter the zone of incompetence. Yeah, you cannot do random things. You have to do some thinking, and I will come with an example. But you have to learn, and you learn by failing, not by making more of the same. Yeah, and this is a misattributed quote from Stalin, Second World War. Yeah, how to win it? Well, make more tanks. Doesn't matter how good they are, as long as they are good enough. Yeah, and that to a certain extent is with drug discovery. You have to do lots of experiments, make them just good enough. Yeah, so we can do this in a little simulation game. So this is a GSK data set. It's 2,500 compounds, and they're like Lego. You have the fixed bit in the middle. You have 50 bricks on the left-hand side and 50 bricks on the right-hand side. So you can make 2,500 different ones, and they did, or most of them. And they measured their activity. Yeah, so here, represented as a matrix and sorted such that you have that island of activity, the top left. So we can say, okay, make this my drug discovery problem. Yeah, so hide all of them. And say, let's find the top 5%, the best ones, and there are 85 of them. And you say, okay, do it in 1 year, 40 weeks, 5 days, 200 days. Yeah, and in a design cycle, 10 compounds, and you have 3 strategies. Just do random, or build, there is a model, use it and pick the 10 best according to your model, or pick the numbers 11 to 20, or 20 to 30, or 30 to 40, and you can do that quicker, yeah, because the design setup cost, the one-off cost, goes down the simpler the method, and the running cost goes down. So I tweaked a bit with these numbers. And what do you get? Yeah? 8 design cycles, number of good compounds found on the y-axis. Yeah? Lesson number 1, models work, yeah, because random is down there. And lesson number 2, the greedy model, yeah, take the numbers 1 to 10, seems to be best, but not by much. Yeah? And this is the average over 10 runs with 10 random starting points. However, if you look at the full picture, you see that greedy only gets 8 cycles, yeah, because they are expensive to run. Yeah, that's when you do a free energy perturbation. Yeah, they last a day. Yeah, some people really throw the boat at the kitchen sink at their design, and that takes too long. Yeah, so greedy works best, but takes too long. Yeah, so in the left-hand side, you can see how other methods get multiple, more iterations in. And on the right-hand side, where I plot it versus time, you can see the difference. Yeah, the next best, the slightly sloppier experiments do beat the greedy experiment, yeah, which is the top of the range, state of the art, but takes too long experiment. A random, yeah, you get there, but it takes an awful lot of time. Yeah, that's not viable either. Yeah, so everything in the design make test cycle, speed of execution matters. Yeah, and maybe it's better not to focus on the AI too much, focus on how to use it best. Yeah. So AI speeds up drug discovery, yeah, and we and competitors and others have, let's say, a track record that shows this, but does not necessarily mean that the success rate goes up. Yeah, faster experiments doesn't necessarily mean better if your design hypothesis was wrong. Yeah, and it also, like any other technology, it takes a while to realize how to best use AI. Yeah, we were quite naive when we started. We definitely worked with a greedy strategy, and it took us a while to realize maybe we should not. Yeah, maybe to look at the whole picture instead of just focusing on one part. And in general, yeah, algorithms and software is getting better. It gets more user-friendly. It's more democratized. Yeah, you can download algorithms and the software. You can have really powerful stuff for free these days. Yeah, so that's, if you start a company, that's not setting you apart from others, because they can download it too. And it's the same with a lot of data to train your models. A lot of that is free as well. Yeah, so if you want to differentiate yourself, you really have to differentiate yourself with your process. Yeah, so not so much the AI itself, but how to use it is your differentiating factor. And that means people, but also organizations and work practices, they need to adapt. Yeah, so the big pharma mindset where things are done in committee, that doesn't work anymore. Well, it does, but not as good as, it's not optimal. Yeah, there are better ways of doing it. Yeah, so as a conclusion, magic does not exist. We knew that, but I'm still very, very positive that those AI-discovered drugs, they will be coming. Yeah, despite all the naysayers and the pessimists, and I'm still an optimist. Yeah, even if you look at the share price of the company I just left, which means I had to sell them at the absolute bottom of the market, but that's, hey-ho. I enjoyed myself massively, and I would do it again, even if I know how it ends. Thank you.
Thank you very much, Willem. I think we have time for some Q&As. There are many. Let's start over there, and then we move to the next slide.
You're off the hook now.
Thank you, Willem. You mentioned chemistry and small molecules progress.
Yeah.
Can you comment on biologics and large molecules?
Well, that's not my area of expertise, but as far as I know, there are AI drug discovery companies who really focus on those as well. And I can see large language models producing, amino acid sequences. Yeah, surely that should work.
Yes.
Yeah.
One other quick question. You mentioned the importance of retrosynthesis.
Yeah.
Do you see the data coming together from different agencies to give us the adequate training sets?
The chemistry has the disadvantage. It's an old discipline, which means a lot of it is in databases, but they are owned by Elsevier or other commercial entities. While biology, as a newer science, I think everything is open and free. Yeah, the Protein Data Bank versus Crystal Cambridge Structural Database. Yeah, free versus paid. And with retrosynthesis, the best data set is probably chemical abstracts, but they treat it like Fort Knox. Yeah, no one gets a look in. Unfortunately, I think as humankind we could move on quicker with that data set.
Philippe Moinjean, I'm a professor of AI in health at Université Paris-Saclay. I think you clearly offered a pretty pessimistic view of the situation, and I think actually it's quite different. I think AI is really transforming very deeply drug discovery. It's very clear that it's accelerating the phase of discovery, which up to now was lasting 5 to 7 years. With AI, there are many candidates now reaching development, I mean preclinical and then clinical development, within 2 years or even less, as opposed to 5 to 7 years. So that's one part. Importantly, those drug candidates are better designed, optimized, and also AI allows to identify therapeutic targets involved in the causality of the disease. There is more than selecting the drug candidates. Just to finish up, it's more a comment, as you understand. There are, as of today, probably several thousands of drug candidates being designed, at least in part, with AI. Up to, I think, 120 have reached clinical evaluation in human being. The most advanced being the one from in silico medicine, which has just completed recently a phase 2 study in idiopathic lung fibrosis, a very complex and difficult disease, and the result turned out to be positive. So I think we should be more positive about AI. I would say transforming for the good the odds of drug candidates, better design, stronger rationale, and of course we should speak as well of this revolution coming up, the new ways with AI to model the disease as it relates to individual patients. In fact, at the end of this scientific days, we have the digital twins, and we have also the virtual patients. This is a huge revolution.
Yeah, I have one little pushback. I mean, I agree with most of what you said, and I also mentioned there was one phase 2 readout that was in silico. But on the other hand, there is a little survivor bias in the sense that they had a hypothesis that a particular target would be beneficial for that disease, but it was not a certainty, and it worked. Well, thank God it did, especially for the patients. But it could just as well not have worked. Yeah, we as in the pharma industry, work on a lot of good rationales why things should work, and so biology is very complicated, and things we look at it with a very reductionist view. One target, one disease, and that doesn't always work. Our compound that was phase 2 ready. Competitors did a phase 2 and they all failed, which sort of proved to us, well, the mechanism is wrong. But the idea behind it was, I think, the right idea at the time. So in silico, hats off to them, they did it. But I would still have said hats off to them if it failed in phase 2. Because the fact is they got there really quick. And that's what you need to do.
There were one question over there.
Yes, hello, Eric de la Fortelle. I'm an investor with Cathay Health, a VC fund. Much as the previous questioner said, I think pharma will incorporate AI drug discovery little by little, as it has incorporated many other waves of innovation. Where I'm more skeptical is the ability of small startup companies using AI for drug discovery to actually thrive. Because people like you, geniuses of informatics, cheminformatics, etc., will come up with a molecule. Maybe this molecule is a genius molecule, but you only know that 7 years later after applying very different skills of medical, clinical development. So it's like asking a hen to climb a tree to get there. And that issue has plagued the whole industry. Do you see a way out of that?
Ooh, well, yeah, the experiment you do is basically you start a drug discovery process, and only in phase 2 you know it worked. Yeah, that's 10 years. And some of the delay is there for a reason. You don't want to dose people with compounds, just like that. Yeah, that would be unethical, to say the least. So I don't see an easy way out. Yeah, if I had, I would start my company and be a billionaire tomorrow. Yeah, if I find an easy way out, I don't think there is one, unless in some future state we have models that are so good that you can have, let's say, a one-shot drug discovery instead of all those design cycles. Yeah, you design the perfect molecule, and if you have a perfect model of the human body, you know it's safe. Yeah, so you can almost straight go to phase 2 if you're there. Yeah, I wish we were there, but maybe in my lifetime. But
Surely.
I don't know. Yeah.